Welcome everyone to today's webinar, comparing immunodeficient models for cancer, immunity, and transplant research. Before we get started, I would like to introduce today's speakers. My name is Cat Chappell. I am a technical information scientist at the JAX Laboratory. I earned my master's degree in integrative genomics in 2015, where my thesis work involved next-generation sequence protocol development and data analysis methods. I have more than a decade of experience developing, implementing, and improving high-throughput laboratory processes. I have been with the JAX Laboratory since January 2021, where I work to streamline high-throughput RNA-seq processes and build protocols for third-generation sequencing platforms. My colleague, Dr. Kaya Ghosh, is a technical information scientist too. She studied microbiology at Cornell University and went on to earn a Ph.D. in molecular parasitology at Rutgers University. Her postdoctoral work at the Albert Einstein College of Medicine in New York City focused on T-cell responses in mouse models of autoimmunity and infectious disease. Prior to joining with JAX, she worked for several years providing consultative and technical flow cytometry data analysis support to clients and corporate partners in the academic, pharma, and clinical diagnostic spaces. Today, we will be discussing the characteristics, benefits, and limitations of some of the popular immunodeficient models available from the JAX Laboratory. We'll also be touching on examples of their uses in research with a focus on the overview of research applications for the really popular model NOD/SCID gamma or NSG. This webinar is intended as an overview of these strains and their research applications. Please be on the lookout for future webinars that will discuss some of these topics more deeply. The search for the perfect immunodeficient mouse strain has been an ongoing process since the late 1960s, when the nude mutation arose spontaneously. With nude mice, researchers could, for the first time, transplant cells from different organisms into the mouse. In the 1980s, the discovery of the spontaneous mutation scid, researchers saw the possibility of human cell engraftment. In the 1990s, the development of immunodeficient mouse models progressed substantially with the creation of the RAG knockout and the IL2RG targeted mutations, and the addition of the scid mutation to the NOD inbred background. The backcrossing of the scid mutation to the NOD genetic background created mice that could engraft human primary cells and tumors, progressing the field further. NOD/SCID mice led to the development of NSG mice. The NSG mouse and its derivatives have led to a preclinical bridge between mouse and humans. Before we can understand how and why we might want to select an immunodeficient strain, we need to know how the components of the immune system work together. There are two main branches of immunity in mice and humans, the innate and the adaptive. The innate branch of the immune system, shown in left in green, can be considered a first-line defense against pathogens and foreign invaders. It contains various components that are hardwired to recognize and respond to diverse pathogens. Innate immunity includes the complement system, which is involved in lysing pathogenic bacteria and cells bound by antibodies. Macrophages engulf dead and dying cells. Granulocytes participate in allergic and inflammatory responses. Natural killer cells are a population of cytotoxic cells that can identify and kill pathogen-infected cells and tumor cells. Dendritic cells are the link between the innate and adaptive immune systems. They do so by presenting antigens to the T cells, allowing them to mount a response. The adaptive branch, shown in blue on the right, includes B cells that produce antibodies and T cells that have multiple functions, including directly killing cells and activating B and other T cells. Cytokines, seen in the middle in the arrows, specifically interleukins, are the secreted proteins that all immune cells use to communicate with one another. Here's a great overview of the popular immunodeficient strains that we will be discussing. You can see them with the red star. Starting on the right, the outbred and inbred nudes, we will go from the least immunodeficient strains to the most immunodeficient strains, ending with the NSG on the right. When considering which immunodeficient strain may be of best use for your research goals, ask yourself the following questions. Do you need certain parts of the immune system intact? And if so, which ones? Which part of the immune system do you need to be non-functional? This could be influenced by factors such as what kind of cells are you planning to engraft, such as are they murine or human cell lines? Are they cell lines or tumor cells? And are you planning to engraft hematopoietic cells? Today, we're going to start with both the inbred and outbred nude mice. Historically, the classic xenograft model discovered in the 1960s was the nude mouse. The nude mutation is a spontaneous mutation in the FOXN1 gene. The FOXN1 gene encodes a transcription factor involved in patterning hair follicles, as well as in the thymus, which is the site of T cell development. Nude mice are athymic, and due to that absence of the thymus, there are essentially no mature T cells. B cell response is diminished because they lack synergy with the T cells. However, as you can see in the diagram, the innate immunity is essentially intact. Some advantages and applications for this model include the engraftment of human and mouse tumor cell lines. They are very well published and characterized, and the hairless phenotype facilitates tumor growth measurements. As we discussed in the previous slide, nude mice still have an intact innate immune system. This means that nude mice can only engraft cell lines and not primary patient tissues like PDX or hematopoietic cancer models. An example of the use of nude mice by Golia in 1994. In this experiment, nude mice were implanted with a 17B estradiol time-release pellet. A few days later, they were injected subcutaneously with the estrogen-dependent human MCF7 breast cancer cell line. The mice were treated with an experimental chemotherapeutic agent intramuscularly every other day for 40 days. As the image suggests, this agent was successful in regressing the tumor after treatment. Because the mice are hairless, it is quite easy to see and measure the nodule with calipers. While a nude mice engrafted with cancer cell line will develop tumors, the tumor that develops does so without the normal microenvironment that you would expect for a human or mouse tumor. Next, we have the BALB/c mice. The scid mutation, which stands for severe combined immunodeficiency, spontaneously arose in the BALB/c mice in the 1980s. Due to the scid mutation, BALB/c scid mice have no functional B and T cells. BALB/c still have an innate immune system intact, and they show complement-dependent cytotoxicity. BALB/c mice can be used as xenografts for some primary cells and tumors. BALB/c scid mice are frequently used for efficacy testing for therapeutic antibodies. While the presence of the innate immune system is required for testing therapeutic antibodies, the innate immune cells, especially the NK cells, can interfere with engraftment of some cells and tissues, such as immune cells or leukemias. The scid mutation makes BALB/c mice more sensitive to radiation and show a higher potential toxicity to genotoxic drugs. BALB/c mice have a high scid leakiness, meaning that the mice may develop mature T and B cells as they age. Here's an example that well illustrates the use of this model in research from Zhang et al. In 2010. Keep in mind that in order for a tumor to grow or metastasize, it requires two things: circulatory invasion, which is where the blood vessels feed nutrients to the tumor, and a functional lymphatic system to pull fluids out of the cells and disseminate the tumor cells to other parts of the body. This study utilized BALB/scid mice engrafted with metastatic human hepatocellular carcinoma cell line HCCLM3, which is known to have a high metastatic potential. The authors looked at the effect of the VEGFR Ig fusion protein, denoted above as the VEGFR31 Ig, and its effect on tumor growth and the metastasis for the HCCML3. As you can see in this figure, the antibody potently blocked the tumor growth over time. The VEGFR31 Ig effectively suppresses the HCCLM3 tumor growth in the BALB/scid mice. This antibody also inhibited metastasis of the tumor to the lungs. You see the reduced lung tumors here, which are also described graphically below that. The arrows indicate metastatic foci. The next strain I would like to discuss is the B6 RAG1 knockout mouse strain. The RAG1 and RAG2 genes are required for B and T development, but act in a different pathway than the PRKDC gene that we discussed with the SCID mutation. RAG1 knockouts, like SCID mutants, have no functional B and T cells. The B6 RAG1 knockout mouse can be used as a xenograft host and to test therapeutic antibodies. Because they are on a C57 Black 6 genetic background, which is the most frequently used background for biomedical research, these mice are frequently used for adoptive transfer from B6 donors and in combination with other mutations or transgenes on the C57 Black 6. An advantage is that the B6 RAG1 knockout mice are radiation resistant, unlike the mice with SCID mutation. The major limitations of the B6 RAG1 knockout mouse is that the presence of the intact innate immune system. Therefore, they are a poor host for primary human cells. In this example, mice infected with neurotropic strains of mouse hepatitis virus develop acute and chronic neurological infections. Susceptible strains of mice infected with the neurovirulent JHMV strain rapidly succumb to acute encephalitis, whereas infection with an attenuated JHMV variant results in a more chronic form of the disease. The disease is largely immunopathological, with demyelination occurring as a consequence of virus clearance. Since RAG1 knockout mice, by definition, do not mount an appreciable T cell response, they are protected. However, transferring splenocytes from immunized mice to the RAG1 knockout mice recapitulates the demyelination one would typically see in immunocompetent mice. The RAG1 knockout mice can be used for adoptive transfer of regulatory T cells. In this experiment, the authors infected B6 RAG1 knockout mice with the MHV strain that will cause immune-related demyelination, which will develop if the mice are reconstituted with either JMHV immune CD4 or CD8 T cells. The FOXP3 positive regulatory T cells and the FOXP3 negative splenocytes were transferred. The mice that received the FOXP3 positive regulatory T cells showed less weight loss and improved clinical outcomes, similar to control mice, and decreased the demyelination than those that did not receive the regulatory T cells. Now let's turn our attention to the NOD-SCID mouse strain, which was generated by JAX scientist Dr. Lenny Schultz by backcrossing the SCID mutation onto the NOD inbred background. The NOD genetic background shown on this slide contributes to the reduced innate immunity independent of the SCID mutation, including the absence of a hemolytic complement and reduction in dendritic and natural killer cells. NOD-SCID mice have that reduced innate immunity due to the NOD background and also have no mature B and T cells due to the SCID mutation. NOD-SCID mice therefore have defects in both their innate and adaptive immunity. The NOD-SCID mice can accept xenografts of some human tumors that do not engraft in the previous strains that I have discussed. They are also capable of engrafting some hematopoietic cell lines. NOD-SCID mice have significantly less SCID leakiness than that seen on the BALB-C and B6 backgrounds. Some considerations in the use of NOD-SCID include that they have a shortened lifespan due to thymic lymphoma that develops around 9 months of age. There is some residual innate immunity, so the natural killer cell function, and the known SCID side effects, which are the radiation sensitivity and sensitivity to the genotoxic drugs. NOD-SCID mice have been shown to propagate medulloblastoma-like tumors, which have been difficult to model in mice. Here, the researchers took neural precursors derived from transformed human embryonic stem cells that exhibit neoplastic features of the human brain tumors and injected them into the brains of NOD-SCID mice. These cells gave rise to tumors with the medulloblastoma-like phenotype in both the posterior and anterior brain. The arrows here denote the presence of neural rosettes, which are found in the medulloblastomas. These arrows point out blood vessels within that tumor. The main takeaway is that medulloblastoma tumors are historically hard to grow in other immunodeficient models. However, their growth in NOD-SCID provides proliferation and infiltration that looks similar to what you would see in a patient. The nod-scid mouse led to the creation of the nod-scid gamma, or NSG mouse, in Dr. Lenny Schultz's laboratory. The NSG mouse strain is a nod-scid strain with an additional targeted deletion of the gene encoding the interleukin-2 receptor gamma chain. The IL-2 receptor gamma chain is a major component of the cell surface receptor for interleukin-2, as well as 5 other interleukins. In the absence of a functional receptor, downstream signaling is blocked from these interleukins, specifically blocking signaling from IL-15, which completely prevents natural killer cell function. As you can see on the right, a lot of things are crossed off. The nod-scid gamma is a severely immunocompromised model. The NSG mice show enhanced engraftment of human hematopoietic stem cells due to the mutation in the SERPA gene in that nod genetic background. They show no scid leakiness, and they do not develop the spontaneous thymic lymphomas. And that means they typically have a longer lifespan than the nod-scid mice. NSG mice can live up to 16 months or longer, which can allow for much longer experiments. The main consideration with the use of NSG mice is that the scid mutation has the side effects we discussed previously, namely the radiation sensitivity and that genotoxic drugs may have a higher toxicity. The NSG mouse strain has become one of the most useful and versatile immunodeficient mouse models to date, with a lot of different research applications. Kale will discuss some of these applications more in depth with you today.
Okay, thanks, Kat. Can you all see my screen? Great. So let's pick up where Kat left off and get a little bit more into depth about the research applications that the NSG mice enable. So a few of the research applications, and they're very diverse, that we'll talk about today are the ability of the NSG mice to serve as a host for primary tumor engraftment, hematological cancers. They're also a great platform for studying basic principles of human hematopoiesis, as well as infectious disease, various autoimmune diseases. We're going to pay some special attention to type 1 diabetes as probably one of the most typical autoimmune disease platforms that folks look at, and some other immuno-oncology platforms, some of which are addressed by JAX in vivo services, which we'll touch on briefly towards the end of the webinar. So let's pick up by talking about the first bullet point here, how NSG mice can serve as a host for primary tumor cell engraftment. So the reason why they're such a great host for primary tumor cell engraftment, or PDX, is that they preserve patient tumor characteristics. So in these histological images, you can see a patient's lung tumor, which is non-small cell fragments here on the left. And these non-small cell fragments were engrafted subcutaneously into the NSG mice here in the top row versus traditional scid mice on the bottom row, which was an earlier predecessor of the NSG mice, as my colleague Kat described. And then the progression of the tumor engraftment was compared head to head after 9 weeks. So as you can see, the tumor engrafted into NSG mice retained a much more typical epithelial carcinoma-like appearance. And this is similar to the patient's biopsy. You can just see as a glance where you compare the H&E staining here in the scid mouse, you just see a lot less nucleation, and it's just a lot. It's much more different than it is in the NSG. And then if you take a look as well at HLA staining for human leukocyte antigen, the scid mouse also has very diminished staining here in brown compared to the panel up at the top, the NSG mouse. Since the NSG is so severely immunodeficient, there is also markedly less infiltration of the mouse white blood cells, which we see by staining for anti-mouse CD45 up here in the top panel, whereas in the scid mouse, you do see significant infiltration of mouse CD45 white blood cells. So in this sense, the tumor microenvironment environment is more faithfully recapitulated in the NSG mouse because you're going to see better engraftment of, for instance, human immune cells if you're choosing to look at this with humanized cells. In the same paper, the authors also compared the patient tumor to the engrafted tumor, and they found a high level of concordance of the engrafted tumor in the NSG mouse compared to the patient tumor, even after 9 weeks of engraftment. So you can see that there's positive staining for cytokeratin to a pretty similar degree between the patient tumor and the engrafted tumor. And this is an important marker for carcinomas. You can also see the proliferation marker Ki67 is also, you see it in both panels here. And so if you take this data together, it suggests that the NSG mice have a remarkable capability in preserving many aspects of the patient tumor characteristics. Because of this remarkable ability of NSG mice to recapitulate the PDX tumor microenvironment, the Jackson Laboratory launched a PDX service where we engraft mice with PDX. We have a variety of PDX samples. Some of them are maintained live in mice, which accelerates their availability. Some of them are cryopreserved in an extensive bank of cells. In either case, we do have characterization data on many of these models, various degrees of characterization. And we do have a webinar coming up in the near future that'll touch on some of these capabilities that we offer through in vivo pharmacology services. But here's just a quick schematic of how this works, where we have the PDX samples either live or cryopreserved, and then we engraft them into donor mice and amplify it up into a greater number of mice that you can use for your experimental cohorts. And then JAX can actually perform a study for you, or we can provide you with the engrafted mice and you can run the study yourself. Let's also talk about NSG mice as a platform for autoimmune research. So type 1 diabetes is a frequently looked at model. In this paper, the authors administered STZ, which leads to synchronous reproducible hyperglycemia in NSG mice, and it mimics the end stage of type 1 diabetes. So here you can see that initially, the mice are clearly hyperglycemic due to the STZ administration. Then here at day 0, the hyperglycemia phenotype is rescued by transplanting beta cells subrenally from a healthy donor. So you can see the blood glucose is coming down. And also it's maintained, the blood glucose is maintained at a healthy level for a few days. Then a reversion to hyperglycemia occurs after a nephrectomy was performed to remove those transplanted beta cells. And then you see a restoration of the hyperglycemic phenotype. One of the most remarkable features of NSG mice is their capacity to support development of a functional human immune system. Now, this is made possible in part by the elimination of host natural killer cells, as this is a cell type that is one of the primary impediments against efficient engraftment of human hematopoietic stem cells. So the hematopoietic stem cells hone to the bone marrow, and they can differentiate into every major type of human immune cell. And this is done through well-characterized precursors. And so this will include cells of the myeloid lineage, like macrophages, granulocytes, dendritic cells, etc., as well as the lymphoid lineage, which includes B cells, T cells, and K cells. And this is a notable achievement because previous attempts to humanize non-SCID mice with human hematopoietic stem cells didn't result in mature T cells developing. In addition, the human B cells that develop in the NSG mouse undergo immunoglobulin class switching. So this is a phenomenon that occurs in B cells across all mammals, and it refers to the fact that B cells first synthesize one type of immunoglobulin at first, but generally there's class switching that occurs and they start making another kind of immunoglobulin. So for instance, IgM is the first antibody produced, and that's important as a first line of defense because that antibody is present in the mucosa mucosa, where the body meets a lot of pathogens. But then as the immune response matures, in many cases, class switching occurs. So for instance, you'll start to see classes expressed like immunoglobulin G or IgG, which is the long-lived, often high-affinity antibody that vaccines aim to elicit, for example. And there's a few other types, of course, which we won't go into today. But the point is that this is a really profound ability of the NSG mouse to recapitulate that class switching that is so important for disease platforms where antibody responses are key. So let's take a couple moments and talk about the experimental timeline for the generation of JAX humanized mice. So we are typically irradiating mice at a young age. And this is for the CD34 cell engraftment. We also do some studies with PBMCs, in which case we're typically not irradiating the mice, but you'll see a variety of protocols out there in the literature. So there's definitely some variation as far as whether or not irradiation is being used and what the dose is. But this is typically what we do here at JAX for the CD34 humanized mice. We're irradiating the mice and then engrafting the cells by tail vein injection at about 3 weeks of age. And then after a couple of months, you start to see human B cells appear and then T cells. So let's go into a little bit more detail about the subsets that you see in these mice. So I like this figure. I'm actually going to show you a white paper a little bit later that goes into a lot more detail, but I like to show this figure because it kind of gives you some broad strokes of what you're going to see in these humanized NSG mice. And this is at 16 weeks because for the most part, engraftment does increase over time for most of the subsets that we're going to be talking about. And here on the left, you can see we're looking at human CD45 positive cells. That's a pan white blood cell marker. And you can see that the T cells are well present and then further divided into subsets of CD4 and CD8. Like I said, there is a paper that'll go into even more detail, which I'll show you a little bit later on. And then you can also see some of these other subsets that have been stained for, including B cells and some of the myeloid lineages as well. And the reason we're having, different data points here is engraftment is donor dependent. So although the results tend to be reproducible from donor to donor, the engraftment rates across donors is variable. And so is the responsiveness of donors to various therapeutic agents you might be testing. So, if you do want to use JAX humanized mice, or even if you're performing humanization in your own laboratory, donor balancing, i.e. trying out, different human donors, especially for your pilot experiments, is a really good idea because you're not sure a priori which donor is going to best serve as a platform for testing the particular agent or whatever hypothesis it is that you're testing based on that variability. So we've touched a little bit on the different kinds of human blood cells that develop in the humanized NSG mouse. But the question remains, how functional are these cells? So here is some semi-quantitative proof of principle data that was collected by the JAX in vivo pharmacology services group that show that humanized NSG mice do in fact show T cell dependent responses. And you can see this in the gross pathology of these mice, which I'm about to describe to you. So in this experiment, humanized NSG mice were subjected to a delayed type hypersensitivity or DTH response. And this involves sensitizing the humanized mice with a chemical called dinitrofluorobenzene, or DNFB, on their abdomens. And then the sensitized mice, when challenged with the same compound applied topically to the ear at a later time point, they display an inflammatory phenotype, and that has been shown to depend on functional T cells. And the readout for this is ear thickness, with a thicker ear indicating swelling in response to this delayed type hypersensitivity. So this graph shows the ear thickness in different groups of mice. And the key point is the green line here is the experimental group, and the other colored lines are various negative controls. So in the green line, the humanized NSG mice were first sensitized to DNFB, and then they were challenged with the compound. And this increase in ear thickness signifies the inflammation that's occurring in those mice. And again, that inflammation is T cell dependent. So this is strong evidence that we're seeing functional T cells in the humanized NSG mice. Like I said, the other lines are various negative controls. The purple line are data points from a humanized NSG mouse that was sensitized and challenged, just like the mice in the green line, but they were then treated with the anti-inflammatory hydrocortisone. So this is just to show that the swelling is indeed due to inflammation and can be dampened by a typical anti-inflammatory of hydrocortisone. The blue line shows a minimal response in humanized NSG mice that were not sensitized to DNFB prior to challenge. And the red line shows that no inflammation occurs when the humanized mice are merely sensitized to the agent, but not challenged. So in conclusion, you can see that the humanized mice do display a specific immune response that appears to be T cell dependent. So I want to add that there's additional functional characterization data available, including at the level of cellular subsets for several of our strains with the NSG portfolio. And a lot of that data will be presented in an upcoming webinar focusing on our NSG portfolio, and that webinar should be announced in the near future. So let's switch gears here and talk a little bit about the use of NSG mice in the field of infectious disease research, one of my favorites. So here's an example where the humanized NSG model was used to validate a promising direction for HIV therapy, which is prophylactic administration of neutralizing antibodies. So in this model, mice were administered an AAV expressing a broadly neutralizing antibody via injection, and the mice were also infused with human PBMCs, and 2 weeks later were infected with HIV. So as you can see on the left graph, the the non-immunized mice show a characteristic loss of CD4 T cells, which is one of the hallmarks of active HIV infection, following infection with HIV. And in the right panel, the humanized mice, the immunized mice, i.e. the B12 here, they didn't display that loss of CD4 cells. The CD4 cells levels stayed much more constant. There wasn't a lasting downward trend in most of these mice, even when the amount of inoculating virus, as you see going down this axis here, was escalated from 1 nanogram all the way up to 125 nanograms. And these mice were followed for several weeks post-challenge. I see this is about 6 to 8 weeks post-challenge. So this set of experiments further validates the PBMC humanized NSG as a useful preclinical model for testing the efficacy of new anti-HIV therapies. There's other applications of human NSG mice in the field of infectious disease research, including EBV, Ebola, dengue, malaria, tuberculosis, and more. And so I would suggest that you click on this link. We'll be making these slides available later to see a list of NSG mice publication references collated by disease area. So here you'll find some of those references in the field of infectious disease, but we've also got a whole lot of other categories that we're collating on a periodic basis. And so this is a great way to just sort of get your feet wet in the literature and browse and see what's out there. So let's just take a couple of moments to talk about the NRG, or the nod rad gamma strain. This is not a strain that we focus on here at JAX in terms of our in vivo preclinical services. We tend to focus more on the NSG strains and derivatives, but I do want to point out the strain because it does have the features that are conferred by the nod background, as well as the IL-2 RG deficiency that my colleague Cat explained in earlier slides. But instead of the scid mutation, it has the RAG1 mutation, which we've also talked about, prevents the development of mature T and B cells. But the RAG mutation confers the radiation resistance that the scid mutation doesn't have. So if looking at higher doses of radiation is somehow important to your study design, the NRG strains might be worth a look. Some of the limitations and considerations include that this is just not as widely used of a strain compared to the NSG strains. So the amount of data out there in the literature is going to be a little bit less. It's a little bit more sparse, but that's not to say that there's not data out there. And we've actually used this strain internally in some instances as well. So we do offer this in our repository. So if this is a strain of interest to you, surely take a look at that as well. And just briefly to talk about the hematopoiesis in NRG mice, the hematopoietic stem cells do engraft efficiently in the bone marrow in these mice as well. And you see differentiation into cells within the myeloid lineage, as well as the lymphoid lineage. Okay, and then engraftment efficiency is roughly comparable to NSG, but please keep in mind that these comparisons are highly dependent on your engraftment protocols, of course. And it also does show very high engraftment of PBMCs. So if you're tending to do experiments using PBMCs, for instance, you might be studying GVHD or doing other shorter course experiments than you typically see used for CD34 mice, that's something to keep in mind as well. So let's spend some time talking about the strain NSG SGM3. Now this strain, of course, it has the NOD, SCID, and the IL-2 receptor gamma deficiencies. And in addition, it has this collection of transgenes, the SGM3. So it expresses human IL-3, GM-CSF, and stem cell factor, also known as KIT ligand. And these together improve the normal human myeloid cell development after hematopoietic stem cell transplantation and promote acute myeloid leukemia or AML xenograft deficiency. So if you're studying liquid tumors, the NSG SGM3 strain is certainly worth a look. I'll also add that even if you're studying other cancers where myeloid-derived cells are important in defining the tumor microenvironment or the general disease phenotype, we often talk about myeloid-derived suppressor cells, for example. The NSG SGM3 strain could also be worth a look to you, even if you're looking at non-leukemia cancers. So a little bit more about the NSG. Um, we do tend to see higher numbers of T cells, including subsets such as CD4 and FOXP3 positive Tregs. Of course, you're going to see higher levels of myeloid cells, which is the reason that the strain was created, and myeloid lineage cells. But I just want to point out that these differences are donor dependent as well. So the degree of difference that you see will depend on the donors you're using. So the differences between the NSG and the NSG SGM3 mice engrafted with the same donor, to be clear, the differences may be more or less pronounced depending on the donor that you're looking at. Okay. So I do see some questions coming up as we're going forward, and there's quite a lot there, and we'll try to answer some of them at the end. But let's see, I'm making a note of them, and I do see some of them are from previous slides, and I'm sorry I missed them because there's so many, but I promise you we'll come back to some of these at the end, so stay tuned. So I'd just like to show you a couple of slides that graphically illustrate the broad trends of engraftment that we talked about. So here's a slide just looking at total human donor cell count, comparing the NSG in green to the NSG SGM3 in magenta here. And so you can see that overall you do see a higher level of engraftment, and we're looking at just total CD45 positive human cells here. And that engraftment does tend to increase over time to a point. And now we'll look at it a little bit more granular. We're looking at the myeloid cells. Again, both increasing over time, especially in the magenta, the NSG SGM3, which makes sense because, again, that was the point of creating this strain, was to get better myeloid cell engraftment. But we also saw better lymphoid development, lymphoid engraftment here, where you're seeing the B cells and the T cells both at higher levels and increasing over time more dramatically than they do in the basic NSG strain. So like I promised, I'm going to point you all to the white paper that goes more into detail about the various subsets that we've immunophenotyped here at JAX in our humanized mice. And so this data that I'm showing you, it's a snapshot of data provided in the white paper from JAX in vivo pharmacology services. This data was collected in collaboration with BD Biosciences. You can click on this link once we make the slides available to access the entire white paper. But here's a snapshot of what you'll see describing how the humanized NSG SGM3 mice show a more robust reconstitution of T cells, and it occurs earlier than it occurs in the basic NSG strain. And so we're looking at two different time points, 10 weeks versus 31 weeks here. And you can see that, in both of the strains, NSG and SGM3, you're seeing an increase in engraftment over time, 10 to versus 31 weeks. But in addition, you're generally seeing more of these subsets in the NSG SGM3 strain, which is the group of nested bars on the right in each plot. So we're looking at CD4 cells, further immunophenotyped into naive CD4, central memory, effector memory, and Tregs. Okay, and as well, the CD8 naive and all of those memory subsets as well. You tend to see more in the SDM3, but remember what I said about the donor dependence. The differences from donor to donor can be more or less pronounced depending on the donor. So I'm going to take a couple of moments and highlight some of the JAX in vivo services that employ humanized NSG mice as a platform. And we do have a webinar coming up. The date hasn't been announced yet, but stay tuned, and that webinar will go into much more detail, but I just want to touch on it briefly. So one of the services that we provide is OncoHu, which is a lot like our PDX service, where we're engrafting human primary PDX tumor samples into mice. But OncoHu is engrafting the PDX samples into humanized mice. So if you'd like to look at the host response to tumors and better recapitulate the human tumor microenvironment with all of the various infiltrating immune cells, the OncoHu platform could be of interest to you. In addition, we have a platform that can evaluate cytokine release syndrome. This is a very dangerous side effect that has been shown to occur, for instance, in the context of CAR T cell therapies. And it's something that can really hinder the advancement of your therapies to later clinical trials, or of course being brought to market. So it's important to characterize the propensity for CRS, and JAX has a unique platform for doing that in humanized mice. And in addition, we can perform efficacy testing of any agents that you might be evaluating for their effect on dampening graft versus host disease. Okay, and there's a number of readouts that we can perform, of course, including body weight and survival. So we're almost wrapping up the webinar here. We keep an eye on the clock. And one of the last things I want to point out is this poster, which describes our comprehensive immunodeficient suite. Now, this is such a large poster that I can't even fit it on one slide, but we do have this poster available for download. And again, once we distribute the slides to you, you'll be able to click on the link and access that poster. It's a really handy guide for just keeping in mind the various immunodeficient mice that JAX offers, and perhaps getting a handle on what might be the best host for your particular applications. So I'd like to close and say thank you all for sticking with us till the end, and we're happy to hear from you either by email or by phone call if you'd like to discuss your needs further.
We do appreciate you tuning in, and as Kay has stated, please reach out to us at micetech@jax.org. We would love to make sure that you get the information you're looking for.